All right, good evening. My name is Hunter, and this is Content Review for Principles of Biology Students 213. Today we are going over diversification of eukaryotes. Can you hear me okay? We'll do a mic check first. My monitor here says it's working. Can anybody hear me? Hooray! Okay, moving right along then. So last week we were really starting to talk about forces affecting allele frequencies in populations and that being a driving force of evolution in populations. And here we're getting to sort of theories around really, really ancient evolutionary processes that led to all of the life that we see on Earth now, and the observations that kind of help us make those form these theories, basically. And evolution having to work with what already exists is these observations led to the conclusion that evolution has to work with what already exists. Things don't just spontaneously appear, as in intelligent design theories, because if they did, we would design people very differently, because our parts do not work very well. So, with that, diversification of eukaryotes. This activity provides an overview of the early evolution of eukaryotic single-celled organisms. The first eukaryotes appear in the fossil record around 2.1 billion years ago. Current evidence indicates that eukaryotic cells arose as a result of endosymbiosis, sometimes multiple endosymbioses among ancestral prokaryotes. The early eukaryotic cells that resulted underwent great diversification to become the seven major groups of eukaryotes. Find the figure in your textbook. Um, this activity is designed to help students begin to understand the vast diversity of the protists, as well as some of the evidence and thought processes that were involved in the development of the endosymbiont theory. And this word keeps coming up, so we're going to go ahead and break it down now. Endo means inside, and symbiotic is organisms working mutually beneficially. Um, Symbiosis, right? Endosymbiont is the engulfing of one organism into another, and that organism being permanently incorporated into that organism's DNA, and then kind of propagating forward uh, in perpetuity and becoming all of the life that we see uh, on planet Earth, which is super cool. All right, so first one, protists have been called a catch-all group. What does this mean? The protists. I'm gonna use a different color. Um, Protists is a paraphyletic group, meaning that it encompasses many of the descendants from a common ancestor, but not all of them, right? So if you have your ancestor and the family tree goes this way, descendants, right? And then you have some branch points. So a paraphyletic group might be, I believe it includes the ancestor, and whatever these descendants are. We'll say descendants one and two. So these would be cousins, right? It includes some of the descendants, but not all. So that's a paraphyletic group. Protists are a paraphyletic group including all eukaryotes except those assigned to fungi, animal, or plant kingdoms. Protists are a paraphyletic group including all eukaryotes except fungi, animal, and plant kingdoms. And I'm capitalizing these, but you do not need to. It's not a convention. Protists range in size from microscopic to macroscopic. Macroscopic meaning you can see it without visual aids like mine or a microscope. Some of the large brown algae can be 60 meters long. Many protists have a characteristic overall form, organelles with distinctive features, or both. And this organelles with distinctive features is important. All eukaryotes have nucleated cells and membrane-bound compartments or organelles. That'll be important later. All eukaryotes alive today, including all living protists, have a nucleus and endomembrane system, mitochondria, or genes that are normally found in mitochondria, and a cytoskeleton. We're going to repeat some of these things, and the repetition is an indication that this is important for being able to identify eukaryotes and distinguish them from prokaryotes and other forms of life. There are more than 100,000 recognized species of protist. Even though the organisms in this group are so diverse, we can recognize some common themes in their evolution, including the evolution of complex cell structure, novel genetic recombination strategies, and complex life cycles. In addition, this group as a whole has great ecological importance. Provide at least 2 examples of protists from chapter 27 in your textbook that illustrate each theme. Now, in an ideal world, you'll have taken a peek at this and picked some examples that resonate with you, that are relevant to your life, perhaps, and that kind of helps you remember them. I have picked a couple that I will stick with that demonstrate each of these themes pretty well. So there are any number of different examples. Pick one that you like. For complex cell structure, we've picked Euglenida. Now, Euglenida contains a light detector or eye spot, contractile vacuole, pyrenoids, and chloroplasts. So we have some kind of light detector, and I believe it's pigmented, so you can actually visualize it. You can see the eye spot. Contractile vacuole. Vacuole is a space inside the cell that can take in usually water from surrounding the cell. Contractile can then compress that and jet it out. I've seen this used as motility. There may be other functions that I don't remember off the top of my head. Pyrenoids, and I actually don't remember what pyrenoids are. Pyrenoids are subcellular microcompartments found in chloroplasts of many algae and in a single group of land plants, the hornworts. Promote photosynthetic CO2 fixation. Cool. That's why I don't remember what they are. Because photosynthetic organisms are not in my wheelhouse, but this is important to know. So it helps with nitrogen fixation related to photosynthesis. Cool. That's one. Paramecium is another good one. Paramecium also contain a contractile vacuole, an oral groove, and cell mouth, so important for ingesting other things. Micro and macronuclei, and cilia. Cilia are those little hair-like protrusions on the outside of the cell that usually help with some kind of motility. Come on now. Okay. So complex cell structure showing off those specialized compartments, membrane-bound compartments inside the cell, and some new specialized structures for things like motility and eating and seeing, interacting with the environment. Right? and that then improving survivability of the organism, which passes on those genetic traits, leading to greater reproductive success and additional evolution. Novel genetic recombination strategies. Um, how do organisms mix up their DNA to form new structures? Paramecium showed up in the previous example, but let's go ahead and use them here. So paramecium has those micro and macro nuclei. So you have your cell, you have a big nucleus and a small nucleus. And the micronuclei function in sexual recombination and reproduction. So two paramecium get together and their micronuclei interact and form new paramecium. The macronuclei function in growth, maintenance, and asexual reproduction. So it's kind of this interesting compartmentalization of genetic material. All right. Another good example is laminaria, which display alternation of generations between gametophyte, which are haploid, and sporophyte, diploid multicellular forms, which gets into complex life cycles. Let's use Plasmodium. Plasmodium has a two-host life history and causes malaria. So Plasmodium is the parasite that is hangs out in mosquitoes and also impacts the blood of humans. Um, and sickle cell in humans is protective against Plasmodium, which actually has to do with not just the shape of the sickle cell and actually being heterozygous, so not actually expressing um, sickle cell disease, the disease state, but being a carrier of the trait changes the chemical environment around your red blood cells enough that plasmodia don't like it. Uh, so it has more to do with potassium concentration. That'll be a fun factoid to whip out. Not going to be on any test that you take for a while, but that is why. So Plasmodium reproduces asexually in its human host and sexually in its mosquito host. So it has a two-host life cycle. Sexual reproduction in mosquito, asexual reproduction in human host. And then ecological importance of eukaryotes. All species of photosynthetic protists in particular, um, in association with things like cyanobacteria, provide the basis for the food webs of all freshwater and marine environments, and is really responsible for the explosion of life that we saw in the Precambrian times, right? So all species. Photosynthetic is important here. In freshwater and marine environments. The reason that this is so important is because a byproduct of photosynthesis is oxygen, uh, right? Plants take up CO2, mush it around, spit out oxygen. And oxygen, the presence of oxygen, and its abundance as an electron carrier in electron transport chains, um, basically set the stage for all more complex forms of life that perform aerobic respiration, which is um, more energetically efficient than whatever came before. Right? All right. You could also talk about absorptive or fungus-like protists and how they aid in the decomposition and recycling of organic matter, especially carbon compounds. Those being extremely important for maintaining carbon cycle, water cycle. In the late 1960s and since, Lynn Margulis provided considerable evidence for the endosymbiotic theory of the origin of various organelles in eukaryotic cells. What is the endosymbiotic theory? Kind of touched on this, but this is uh, the theory that proposes that eukaryotic cells, eukaryotes like you and me, originated when ancestral prokaryotes, most likely an archaeal species, archaea are more closely related to eukaryote than they are prokaryotes, but we'll get there. Um, an ancestral prokaryote engulfed another formerly free-living prokaryotic cell and enclosed them in vacuoles, and those endosymbionts became major eukaryotic organelles. So, endosymbiotic theory proposes ancestral prokaryote engulfed another formerly free-living prokaryotic cell, enclosing ing, come on, enclosing it in vacuoles. Endosymbionts, the organism inside and the one that engulfed it, or excuse me, the one that was engulfed, became major eukaryotic organelles. And we're about to get a little bit more into that. Which two eukaryotic organelles were proposed to have arisen as endosymbionts, and what evidence did Margulis present to support each organelle as an endosymbiont? You know this. Mitochondria were proposed as evidence or presented as evidence for endosymbiont theory for a couple of reasons. It is double membrane bound. And that double membrane is important, which we will get. Yeah, no, we're going to talk about that right now. So mitochondria are bounded by two membranes. They contain their own circular DNA, which I'm going to do, we're going to do these in blue. They contain their own circular DNA, which is different from our DNA, which is linear when it's condensed into chromosomes, right? And 70S ribosomes. Some of the inner membrane proteins are more similar to those of prokaryotes than eukaryotes. This is important. So this membrane that I have drawn in red, and with the associated proteins that I've drawn here, are more closely related to eukaryotes, while those on the inner membrane are more closely related to prokaryotes. And this is evidence that the prokaryotic DNA, which form the mitochondria, are more closely related than to the outer membrane. This is also just good evidence of that, like, engulfing having occurred. If you, do I want to draw this? I kind of want to draw this. Endocytosis. You have what will become a mitochondria outside of the cell. It encounters this membrane, or this other cell, still prokaryotic, creates an infolding of the cell membrane, and incorporates it into its cytoplasm. And that's that double membrane, or that is that evidence of that double membrane. On the outside of this vacuole, should not be blue, is going to be studded with proteins that look more like the cell that's on the outside than the cell that it descended from. I'm going to keep that, just make it really tiny. Here we go. Has a double membrane. Mitochondria have their own circular DNA, distinct from the rest of the organism. And inner membrane proteins are more similar to those of prokaryotes than to eukaryotes. And this is true for your mitochondria too. Your mitochondria have circular DNA, and you get all of your mitochondrial DNA from your mom. Next up, very similar, is chloroplasts. So if mitochondria are the powerhouse of cells in our bodies, chloroplasts are the powerhouses of plant cells, right? They are very, very, very similar in structure and function. Just one second. Please be the right notebook. Yes. Yes. That one. Nope, that one. There we go. I have my old drawings here that I like to refer back to in my notes. In fact, yeah, let's just do this. No, let's not. Okay. Chloroplasts are also double membrane bound. We'll do this one in green, just because. And within that inner membrane, you have those stacked photosynthetic structures. They also have circular DNA and, come on, proteins on the inner membrane are more reminiscent of prokaryotic proteins than are the proteins on the outside membrane. So again, we have that double outer membrane. They have their own circular DNA, as do the mitochondria. Um, oh, 70S ribosomes are also common between them. And inner membrane proteins more similar to those of prokaryotes than eukaryotes. Fantastic. Secondary endosymbiosis, simply having occurred after the first. So how do we decide which one came first? Oh man, like that formatting looks funny. That is because it is funny. The chloroplasts of Disacristata, chromalveolates, and rhizaria are all thought to have arisen as a result of secondary endosymbiosis. What is secondary endosymbiosis? Said to have occurred when a heterotrophic eukaryote engulfs a photosynthetic eukaryote and then becomes an endosymbiont. Secondary endosymbiosis is said to have occurred when a heterotrophic trophic eats hetero other things, gets its energy by eating other things. A heterotrophic eukaryote engulfs a photosynthetic eukaryote, which then becomes an endosymbiont. Organism that eats other things eats a photosynthetic eukaryote, which is getting its energy from the sun, and they become one organism. What evidence supports this idea of secondary endosymbiosis? Remember before, we were talking about, let's say we have that photosynthetic cell with its chloroplasts, and around it was a membrane of the ancestral organism that ate the first one. And then we had the other eukaryotic organism eat that one. So basically, the presence of four chloroplasts surrounded by four, count them, membranes, serves as evidence of secondary endosymbiosis. Carries out photosynthesis and its own plastids. Cool. Structurally, Giardia lamblia lacks complete mitochondria and was once thought to be an example of what the earliest living eukaryotes may have looked like. I strongly encourage you to look up what these organisms look like on the CDC page. Pretty cool looking, very iconic. They will ruin your day though if you happen to catch them. If Giardia is similar in structure to the earliest living eukaryotes, what does this imply about the order of evolution of the various eukaryotic organelles, that is, nucleus, cytoskeleton, mitochondria, and chloroplasts? So Giardia has several flagella for moving around and double nucleus containing its genetic material. Right? However, it contains no plastids and no functional mitochondria. No functional mitochondria. Giardia's structure indicates that the double nucleus in eukaryotes evolved prior to the endosymbiosis of mitochondria or chloroplasts, if this is indeed the case. So presence of double nucleus but no mitochondria or plastids for performing either aerobic respiration or photosynthesis would suggest that the double nucleus um, in eukaryotes evolved prior to endosymbiosis of either the mitochondria or the chloroplasts. We have since discovered that the diplomonads, such as Giardia, and the parabasalids, the other group of excavates, have modified mitochondria. What types of mitochondria do they have? Excavates contain nuclear genomes containing genes that are normally found in mitochondria or unusual organelles that appear to be vestigial. Normally found in mitochondria or unusual organelles that appear to be vestigial, meaning non-functional mitochondria. This is where molecular biology really made the case for um, and provided better evidence for a specific progression in the evolution of life. Um, we would not have known necessarily without this molecular evidence that excavates like Giardia contained genes for mitochondria before that technology was available. We could only go by what we could observe. We didn't see mitochondria. And so because of that, we had to conclude that the double nucleus evolved first, when in fact, mitochondria had to have evolved prior to the double nucleus because those genes were present in that genome, even though they are not expressed. What does this evidence indicate about the evolution of the various eukaryotic organelles? These observations support the hypothesis that the ancestors of excavates had mitochondria. Had mitochondria. But these organelles were lost or reduced over time. So whatever strategy that this ancestral organism had for producing its own energy worked better than the endosymbiont that was the mitochondria, and so it evolved out. Or there was some other pressure that meant that having a mitochondria was no longer beneficial or was harmful. Uh, Going back to last week's exercise and why we would lose that. All right. The metabolic pathways of organisms living today evolved over a long period of time, undoubtedly in a stepwise fashion because of their complexity. Considering everything you have learned to date about the evolution of life on Earth, put numbers 1 through 5 in the following blanks to indicate the order in which these organisms might have evolved and explain your arrangement. Note that some steps happen at the same time and will be part of the same stage. So, little test taking strategy here. Looking at these metabolic pathways, we see photosynthesis together, we see electron transport together, Krebs cycle, and then glycolysis is here by itself. Glycolysis, glyco, sugar, lysis, breakdown, simply means the breakdown of simple sugars. For the endosymbiont theory to have taken place, you had to have a heterotrophic organism consuming the other. And this metabolic pathway, glycolysis, is the most simple on this list, at least, right? So this is probably our first one. We could also get here by looking at what organisms they're talking about, prokaryotes versus eukaryotes. So all of our eukaryotes had to come later. Um, yeah. So for first one, glycolysis in prokaryotes. Um, It's found in all eukaryotes and many prokaryotes, and it takes place in the cytoplasm and can occur in the absence of oxygen, and this is important. Um, I talked a little bit about photosynthetic eukaryotes being responsible for the explosion of life that came after because aerobic respiration wasn't a thing yet. Um, so glycolytic prokaryotes. Next up, we're still on prokaryotes, but we've got prokaryotes performing photosynthesis. I'm just going to put a little squiggle here. And prokaryotes performing electron transport. Which came first, or did they happen together? Photosynthesis creates a lot of oxygen as a byproduct, enough that we were able to saturate the planet with it. And using that oxygen for electron transport was then more possible, right? So prokaryotic photosynthesis requires electron transport and produces oxygen as a byproduct. Neither the Krebs cycle nor electron transport can occur in the absence of oxygen. Both of those things had to happen at the same time. Electron transport is required to convert NADH to NAD plus, which is important for a lot of these metabolic pathways or for the Krebs cycle. Because glycolysis produces 2 net ATP and 2 NADH, the addition of electron transport represents an advantage here. Organisms can then gain 8 net ATP from glycolysis plus electron transport. Yeah. Um, Next up, we still have a couple prokaryotic options here. Prokaryotes. Yeah, yeah. Krebs cycle cannot occur without a mechanism to convert NADH to NAD+. Electron transport must have evolved before the Krebs cycle. It probably evolved in prokaryotes first, and then eukaryotes capable of glycolysis, Krebs cycle, and electron transport as oxidative phosphorylation are thought to have evolved due to endosymbiosis in an amitochondrial organism and an aerobic bacterium. So those are thought to have evolved around the same time. And finally, eukaryotes capable of performing photosynthesis. Some of the eukaryotes with mitochondria are thought to have undergone another endosymbiosis with a cyanobacterium to produce chloroplasts in photosynthetic eukaryotic cells. I think that's about all I got. Simplest first, electron transport and photosynthesis emerging at about the same time in prokaryotes. Photosynthesis in the presence, excuse me, electron transport with oxygen requires oxygen. An explosion of oxygen presents an advantage. Krebs cycle evolves. Eukaryotes performing electron transport and the Krebs cycle come next. And then secondary endosymbiosis happens last. Cool. That's all I've got for you this week. Thank you for your attention. Make sure you get those worksheets in by Friday. I know midterms are coming up. Mine are on Monday, so I will be putting my head down over the weekend. But don't forget to step outside, enjoy the sun, take care of yourself, stay hydrated. I'll see you guys next week.